Well, thank you all for the opportunity to come here today, and thanks for sitting in to listen to some of my research. I see some familiar faces out here that have heard me speak about this topic, and so I'll be providing some updates to the research for those who have listened to these talks before. So today I'm going to be speaking kind of following along the lines of nitrogen impacts on water systems. But I'm going to talk about those impacts below ground. We've heard about these tipping points that can lead to hypoxic zones in the Gulf of Mexico, as well as these harmful cyanobacterial and algal blooms in surface waters. But in the ground, similar processes can be ongoing, although it's going to be slightly different to the impacts that they have. And of course, this is also driven by life that lives below ground. And before I begin, I do just want to acknowledge especially the USGS 104G program for supporting much of this research, and of course the graduate students who have done much of the research that I will be presenting today, and they're denoted in bold here. And of course, collaborators at the University of Nebraska, as well as collaborators at the USGS and the Department of Energy. So we know that nitrate groundwater contamination has been linked to anthropogenic activity, especially that from applied nitrogen to the surface. And this occurs through processes which lead to nitrate leaching down to these shallow water tables, very common here within the state of Nebraska. This is not only just restricted to Nebraska, but of course, we look at the High Plains Aquifer. This occurs throughout the High Plains Aquifer as well. So, and this may occur from, of course, ammonium applications on the surface, or even organic nitrogen that might result in ammonium production. But of course, we have the end result of nitrate that does get produced. Now, nitrate tends to be pretty stable in environmental systems if you have oxic conditions, although there are some processes which have indicated that, yeah, even if your oxygen is present, you can have some nitrate reduction. But much of the nitrate reduction occurs when you go into these anoxic systems, some environments in which are devoid of oxygen. And this is where you get your denitrification reactions. But what's important to remember is that through this denitrification reaction, there's always the production of nitrite, a reactive intermediate. And this becomes important. And in the subsurface, we know that there are microorganisms that do play a critical role in nitrate reduction. If we go out and we look for these organisms in these subsurface systems, we can find that they're very abundant. 10 trillion cells per gram of sediment is what we can find. So a gram of sediment is about the tip of your pinky finger. So these nitrate-reducing bacteria are very abundant, so they have the potential to be playing catalytic roles in the subsurface environment. And when we measure their activity, specifically targeting these organisms, we do see this production of nitrite as a reactive intermediate through these reduction processes. So what does this mean? Well, there are other chemical species that are present in the subsurface that are naturally occurring. One of those is uranium, and so I'm going to be talking to you today about this interaction of nitrate with uranium. And potential, and of course the upshot, and I'll tell you now, is that we think that nitrite is actually playing a significant role. So it's the reduction of nitrate to nitrite that might be actually driving conversion of uranium to a mobile state. And why is this important? If we start looking across the United States at groundwater concentrations that are uranium in groundwaters throughout the United States, we do see that there are regions in which uranium does exceed that maximum contaminant level of 30 ppb. And of course, in some of these regions, it isn't direct anthropogenic inputs of uranium into the system. There isn't a known direct anthropogenic input. And we could say that's very specific for here in northeast Nebraska, is that we don't have a known direct anthropogenic source. So perhaps it's a geogenic source that could potentially be contributing to these high uranium concentrations. So it's important to recognize uranium geochemistry and how uranium will potentially behave. So most often uranium is deposited in these subsurface environments as a reduced mineral. So when it's reduced in this anoxic state, it tends to precipitate as a solid phase mineral and be solid associated. So it cannot move with the water. Whereas when it becomes oxidized, it will bind with other ligands, such as carbonates, and is going to be highly mobile. And when uranyl species do bind with carbonates, they tend to not bind to a sediment matrix, so they become more mobile. Oops. So what we know is that there are microorganisms that can actually breathe uranium. So they eat carbon like you and I will be eating our lunch shortly and get their carbon and energy from. But they're going to breathe that uranium-6, and when they do this, they convert it to this solid state. So they can actually precipitate it. So they exhale out a mineral, so to speak. So some of the challenges are in these natural systems where you have this reduced environment, where you have the uranium-4 that is present, ideally, keeping it at that natural state becomes important. So introducing an oxidant into that system can start to create a challenge. So when uranium is in that oxidized state, it can bind, as I mentioned, it can combine with ligands. And it has been recognized that carbonates can drive uranium mobilization. However, an alternative hypothesis, in addition to not only carbonates, is that of the oxidative dissolution of these uranium-4 bearing minerals. And we know that microorganisms can actually drive this. It might simply be those nitrate-reducing bacteria that can produce that reactive intermediate of nitrite, which can react with the uranium and change it to that mobile state. Alternatively, you have these bacteria that can eat iron, and they precipitate out rust, and the rust itself is an oxidant and is reactive and can oxidize the uranium. However, that iron oxide can also be a sorbent, so it's not always as reactive. But there's also bacteria that can eat the uranium, and when they do that, they change it to a mobile state. So there are a lot of abiotic and biological reactions that can potentially drive uranium mobility in these environments that are driven by nitrogen. So if we start to look at what data is available to look at these differences or groundwater quality measurements within the High Plains Aquifer and the Central Valley Aquifer, we start to see that alkalinity and calcium are correlated with groundwater uranium concentrations, which is well recognized. And we do know that there'll be uranyl calcium carbonate species that will form, and those do tend to be very highly mobile in groundwaters. But we also see that oxidants such as nitrate, iron, and manganese are also correlated with high concentrations of uranium in groundwater. And it's important to recognize, too, that nitrate will also react with iron and manganese, so there can be this co-issues going on. So if we look at the distribution of nitrate and uranium within these groundwater systems, we do see that there is an overlap where we have high nitrate concentrations in the dark blue and higher uranium concentrations which are in the maroon. And we see this purple color begin to appear. So if we look for spatial correlation, we can find that there are regions in which high nitrate concentrations are strongly correlated to high uranium concentrations in groundwater as well. So these correlations are all well and good, but we all know that correlation doesn't necessarily equal causation. So in an effort to begin to examine whether or not nitrate does indeed drive the mobilization of naturally occurring uranium, we selected a field site out near Alden, Nebraska, in order to be able to conduct a series of experiments. Now this aquifer we have measured18 as high as 300 ppb within the groundwater. That's 10 times above the MCL. Nitrate has been measured at 30 ppm, which is 10 times, or 3 times above the MCL. So but we wanted to use samples in which we know were reduced, still had relatively lower uranium concentrations in the groundwater. So we went to a region where we thought would be reduced and would have uranium-4 present within those sediments. So we collected subsurface cores via geoprobe coring, and we maintain the anoxic atmosphere. As soon as those cores come up from the ground, we immediately place those cores under an argon atmosphere to make sure that oxygen does not alter the redox state of those reduced species in the subsurface. We then bring it back to the lab and immediately process everything in an anaerobic glove bag to make sure we do maintain those anoxic conditions and characterize them. And I'm going to specifically talk about the geochemical characterization we've done. So from the system, what we've been able to identify is regions in which we have very high concentrations of uranium within the sediment. Well, and I guess high relatively speaking to the other regions within this system. When we conducted Zane's analysis, and this was in collaboration with John Barger's group at the Stanford Light Accelerator Laboratory, they determined that half of the uranium is present as uranium-4, and the other half is present as uranium-6. So indeed there are regions in here which we do have high uranium-4 concentrations. So we use these sediments and we put them into upflow column reactor experiments within the anaerobic glove bag. So you're looking at a shot of a series of column experiments taken right through the inside of the glove bag. And one of the things that we wanted to do in these systems is make sure, because we know that there's 50% uranium-6, to drive that off of the surface of the sediments by having a bicarbonate buffered medium and influent that we put into these column reactors. So that would drive off the uranium-6. And then we'd add nitrate into the system to stimulate the mobilization of naturally occurring uranium. And I want to point out, we do not add any additional uranium to these experiments. What we do see is at the onset, we do see that there is this carbonate-mediated release of uranium from the sediments. And that's consistent with the published literature and what we would expect based on the uranium geochemistry. And as soon as we add the nitrate, we begin to see nitrate reduction to nitrite, and we begin to see the release of uranium in the effluent. So we see a response that directly correlates to this addition of nitrate in the system. We also followed cell numbers within the experiment. We see this oscillation in cell numbers, which seems to pattern the nitrate reduction as well as some of the uranium release. So we began to think that the microorganisms are playing a primary role actually controlling these processes. But the question is it actually microbially mediated? So we repeated the experiment, but this time we wanted to inhibit microbial nitrate reduction. And so by doing so, we added a chemical called sodium azide, which essentially it's an electron transport inhibitor. It stops the breathing, if you will. And so if the organisms can't respire, they cannot reduce nitrate. So indeed, what we end up seeing is similar to what we saw before. The initial onset, we do see a carbonate-mediated release. We add the nitrate in the system in which nitrate was added to the live sediments. We see uranium release, but we do not see the uranium release when azide is added, which further supports that, yes, this is a microbially mediated process. And again, we're seeing a reduction of nitrate to nitrite. In this case, we also saw some denitrification that is occurring. So together, these results reveal that, yes, desorption is playing a role in releasing uranium into these systems, but also biological dissolution of uranium can also play a potential role in increasing groundwater uranium concentrations, which brings us to this question of, well, do we have to maintain this reduced environment to prevent uranium mobilization? Well, if we go back to the metadata and we start to look at where are these regions into which we saw a strong correlation, it would actually suggest that no, necessarily you don't have to have no nitrate within the system, but perhaps there might be a tipping point, and that tipping point might be closer to the MCL that's already established. So we had conducted a study, another column study, in which we had basically no nitrate or nitrate coming through an upflow reactor system. And what we ended up seeing is that with low concentrations of nitrate, we actually saw uranium retention within the column. Whereas when we omitted uranium or omitted nitrate from the system, we saw a loss or mobilization of uranium from the system. So what this indicated to us was that perhaps there is this tipping point where nitrate might, in an oxidant into these reduced systems, could actually start stimulating organic matter decomposition, which leads to uranium reduction and retention. We look at the iron data, we see a similar pattern, where when we have nitrate in the system, we lose iron. And that shouldn't happen because we would expect iron oxide precipitation in the system, but that's not what we see. So it's very consistent with what we would expect within these systems, for there to be some stimulation of reducing conditions. Unfortunately, I don't have time. We actually have some in situ evidence from a milling impacted aquifer in Colorado, which further supports that with the influx of oxygen into that system. So what we think is actually happening with regards to the tipping point is at low concentrations, when you have nitrate into the system, you have oxidation of organic carbon, and that frees up more organic carbon so that these organisms that can use the uranium-6 will eat the organic carbon while breathing the uranium to precipitate out the reduced iron. But also, we have to also keep in mind that some of these iron oxidizers that can oxidize that iron, precipitate out rust, could potentially serve as sorbents. So we conducted a series of experiments. Oops, I'm going to cancel that. Okay. A series of adsorption experiments in which we adsorbed uranium to reduced sediments. So we basically did not let those reduced sediments get exposed to oxygen at all. Where the other series of experiments was taking those same sediments and air oxidizing them to precipitate any iron oxides. And we do see a difference in the amount of uranium that's absorbed to the surface. So in collaboration with Kate Campbell at the USGS and Boulder, and of course now at the Federal Center, we were able to account for this difference in absorption simply by the precipitation of ferrihydrite, an iron oxide mineral. So iron oxides can also play a role in potentially retaining uranium. So perhaps even those two together. So while we might see uranium mobilization with high concentrations of nitrate, perhaps even low concentrations of nitrate might be somewhat helpful in retaining uranium in that system. So this would indicate that there are tipping points with regards to uranium mobility and other mechanisms that might be at play in retaining uranium in these systems. So with that, thank you all, and I will be happy to take questions after the next talk, right?